Good morning. Today, myself Priyanka from Bsc Chemistry Honors Final Year. We are here to discuss an organic chemistry chapter, and the chapter name is proteins. So, let's start the topic. Proteins. Greek protein means first, are extremely complex natural polymers and are rated first amongst the organic compounds essential for growth and maintenance of life. They are present in almost all living cells, being found in almost every part of every plant and animal. In human beings, they are the main constituent of muscle, skin, hair, nails, tendons, arteries, and connective tissues. In nutshell, each living cell is made up of thousands of different proteins. The proteins present in different plants and animals, and even the proteins present in different tissues of a particular part of plant or animal are different from one another in the composition and biological functions. However, all proteins contain the same elements, that is carbon, hydrogen, oxygen, nitrogen and sulfur. Some of these may also contain phosphorus, iodine and traces of metals such as iron, copper, zinc and magnesium. All proteins on hydrolysis give alpha amino acids. About 26 alpha amino acids have been isolated from the hydrolysis of proteins, but the number of proteins obtained from these amino acids is very large because of the fact that there is no restriction on the number, types, and specific sequence in which alpha amino acids could be present in a protein molecule. Plants alone can synthesize proteins from CO2, water, and inorganic nitrogenous compounds, but animals mainly depend upon plants for their protein requirement. Classification of proteins. Proteins have been classified in number of different ways. The two most widely used systems of classification are discussed below. On the basis of molecular structure, proteins have been classified as fibrous proteins and globular proteins. Fibrous proteins. Side, these proteins consist of linear thread-like molecules which tend to lie side by side to form fibers. The polypeptide chains in them are held together usually at many points by hydrogen bonds. As a result, intermolecular forces of attraction are very strong and hence fibrous proteins are insoluble in water. Further, these proteins are stable to moderate changes in temperature and pH. Fibrous proteins serve as the chief structural material of animal tissues. For example, keratin in skin, hair, nails, and wool, collagen in tendons, fibrin in silk, and myosin in muscles. Second is globular proteins. The polypeptide chain in these proteins is folded around itself in such a way so as to give the entire protein molecule an almost spheroidal shape. The folding takes place in such a manner that lipophilic non-polar fat soluble parts are pushed inwards and hydrophilic parts are pushed outwards. As a result, water molecules interact strongly with the polar groups and hence globular proteins are water soluble as compared to fibrous proteins. These are very sensitive to small changes in temperature and pH. This class of proteins include all enzymes, many hormones such as insulin from pancreas, thyroglobulin from thyroid gland which produces the hormone thyroxine, hemoglobin which transports oxygen from blood to muscles, fibrinogen which is converted into the insoluble protein fibrin and thus causes clotting of blood, albumin in eggs, etc. etc. Each globular protein has its own characteristic geometry which results through the interaction between different sites in the same polypeptide chains. These interactions are usually of four types: disulfide bridging, second is intramolecular hydrogen bonding, third is van der Waal interactions, fourth is dipolar interactions. Globular proteins may consist of a single polypeptide chain or may consist of several chains called subunits or protomers. Each subunit has its own three-dimensional geometry The various subunits are then held together by secondary forces such as hydrogen bonding, van der Waals interaction, etc. to give the molecule its overall spheroidal shape. Second is on the basis of chemical composition or the nature of products they give upon hydrolysis. Proteins can also be classified as follows. First one is simple proteins. These proteins on hydrolysis give only alpha amino acids. For example, albumin in the white of eggs, gluterin in wheat, oxygenin in rice, keratin in hair, nails, bones, etc. Second one is conjugated proteins. These proteins on hydrolysis give a known protein portion in addition to the alpha amino acids. This known protein portion is called the prosthetic group. The main function of the prosthetic group is to control the biological functions of proteins. Depending upon the nature of the prosthetic group, the conjugated proteins are further divided into many number of different types as shown in table 6.2. Here, table 6.2 shows the types of conjugated proteins. One column contains name of the protein and the second one contains the prosthetic group. For nucleoproteins, prosthetic group is nucleic acids. Glycoproteins, the prosthetic group is sugars. For lipoproteins, the prosthetic group is lipids such as lecithin. For phosphoproteins, the prosthetic group is phosphoric acid residues. For chromoproteins, the prosthetic group is some coloring matter such as red colored protoporphyrin. Third one is derived proteins. These are the degradation proteins, products obtained by partial hydrolysis of simple or conjugated proteins with acids, alkalis or enzymes. For example, proteoses, peptones and polypeptides. Proteins goes to proteoses, then to peptones and then finally polypeptides. Then we will discuss about general characteristics of proteins. Some important characteristics of proteins are summarized below. First is elemental composition. The elements generally present in proteins are carbon, hydrogen, oxygen and nitrogen. Their approximate composition is almost always C is 53%, hydrogen is 7%, oxygen is 23% and nitrogen is 16%. Sulfur and phosphorus when present occur to the extent of 1% and 0.5% respectively. Second characteristic of proteins is high molecular mass. Proteins have high molecular masses ranging from 10,000 to over 50 millions. The reported molecular masses of some proteins are egg albumin is 34,000 to 44,000, milk albumin is 35,000 to 40,000, where hemoglobin is 65,000 to 70,000 and casein ranges up to 75,000 to 1 lakh. Third characteristic is physical state. Proteins are generally colorless, amorphous solids having no definite melting points. However, some of them have been obtained in the crystalline state. They are usually colloidal in nature. This property of proteins to readily form colloidal solutions with water has been used to separate them from soluble crystalline salts by the process of dialysis. Fourth characteristic of proteins is amphoteric nature. Like alpha amino acids, proteins also exist as Zwitter ions or dipolar ions and hence react both with acids and bases. Fifth is isoelectric point. Like alpha amino acids, each protein has a characteristic isoelectric point, that is pH, at which its ionization is minimum and is least soluble. For example, casein has an isoelectric point of 4.6. This property of proteins is often used for their isolation. Third characteristic is optical activity. All proteins are optically active and further all of them have L configuration since they are made up of L amino acids. Seventh characteristic of proteins is hydrolysis. All proteins can be hydrolyzed by acids, bases or enzymes. The hydrolysis proceeds slowly and in stages, ultimately giving alpha amino acids. The sequence of hydrolytic products is shown below, in which each hydrolytic product is less complex than the preceding one. Proteins to proteoses, proteoses to peptones, and peptones to polypeptides, then to simple peptides, and then finally alpha amino acids. Conjugated proteins also yield products other than peptides and alpha amino acids. Then we will discuss about color tests of proteins. Proteins give a number of color reactions which are often used for their analysis. Some of these reactions depends upon the presence of particular amino acids in the proteins, while others are due to the presence of peptide linkages between different amino acids. Some important tests are described. Biological functions of proteins. Proteins constitute an indispensable part of our food. These are required for building new cells and repair of worn out ones. Obviously, children and expectant mothers require more proteins, but before they can be utilized by the body, they must be hydrolyzed to the constituent amino acids. Hydrolysis of proteins begins in the stomach where HCl causes denaturation of proteins. The denaturated proteins are then hydrolyzed by the enzyme pepsin into lower molecular mass polypeptides. In the intestine, the enzymes trypsin and chymotrypsin complete the hydrolysis to alpha amino acids. The alpha amino acids are then assimilated in the bloodstream and are carried to the cells in the various parts of the body. There, some of the amino acids are converted back into specific proteins which are required by the body for its growth and maintenance, while others get burnt as fuels to supply energy. Thus, proteins are being constantly broken and resynthesized in the body, and this continuous dynamic process helps to fulfill the needs of the body. Having discussed above as to what happens to the proteins in the body, let us now discuss some of the important biological functions of proteins. Some of the important biological functions of proteins are listed below. First function is structural materials. One of the most important function of proteins is that they serve as the chief structural material of animal tissues. Structural proteins are approximately one half of the body's total protein content. Some common examples of structural proteins are keratin in skin, hair, nails, wool, horn, feathers, etc. Collagen in tendons, myosin in muscles, fibrin in silk, etc. Transport agents. Certain proteins serve as transport agents or delivery vents in the body. They transport the cellular necessities such as oxygen, metals, fatty acids, hormones, etc. For example, hemoglobin present in blood transport oxygen from lungs to all the parts of the body. Third is enzymes. These are globular proteins which act as biological catalysts. Their action is very specific and highly efficient. They catalyze the reaction in aqueous solution without any change in temperature or pH. They can bring about breakdown and synthetic reactions much more rapidly in the living organisms at body temperature, and this then is possible in the laboratory even under drastic conditions. For example, the enzymes carbonic anhydrase present in the red blood cells can catalyze the decomposition of over 36 million molecules of carbonic acid, that is H2CO3, to CO2 and H2O in the reverse reaction, that is the conversion of CO2 and H2O into carbonic acid, is catalyzed by the enzyme carbonic hydrase. This reaction helps in maintaining CO2 level in the body fluids and tissues. Similarly, enzymes pepsin, trypsin and chymotrypsin catalyze the hydrolysis of proteins into amino acids in the digestive tract. The enzyme ptyalin present in saliva catalyze the hydrolysis of starch into maltose during the chewing of food in the mouth. The enzymes maltase, lactase, sucrose, and amylases catalyze the hydrolysis of di and polysaccharides into monosaccharides. The deficiency of even one enzyme in the body can either slow down or prevent a particular reaction in the body and thus cause some serious disease. For example, the congenital disease phenylketonuria is caused by the deficiency of the enzymes called phenylalanine hydroxylase. Similarly, the disease albinism is caused by the enzyme tyrosinase. Fourth is metabolic regulators. Some globular proteins regulate metabolic processes. For example, the protein hormone insulin from pancreas maintains glucose level in the blood. The glycoprotein thyroglobulin from thyroid gland helps in the synthesis of hormone thyroxine. Fibrinogen, which is converted into the insoluble protein fibrin and thus causes clotting of blood. Nucleoproteins, constituents of genes, carry genetic information from parent to the offspring. Fifth is antibodies. These are proteins which defend the body against the invasion of foreign organisms. When an infectious species, virus, bacteria, or a foreign protein enters the blood, it releases foreign substances called antigens. In response to these antigens, the body proteins produce specific proteins called antibodies. The antibodies thus produced then specifically combine with the antigens which induced their synthesis, thereby protecting us from disease. Gamma globulins present in blood plasma are example of antibodies. From the above discussion, it is apparent that proteins perform a variety of biological functions. Therefore, they constitute an indispensable part of our food. Here our topic proteins ends. Thank you.